So last time we talked about prokaryotic microbes, the very simple unicellular organisms. This time we're going to talk about eukaryotic microbes. And these guys can be very simple, but they can also be very complex in terms of their form. But the eukaryotic cells themselves are much more complicated than prokaryotic cells. So let's find out a little more about them. So if you remember back to our very first lecture, we had a timeline and prokaryotic cells came along before eukaryotic cells. But they both evolved from this precursor cell called the last common ancestor. And this cell wasn't considered to be prokaryotic nor eukaryotic, but that it did give rise to both prokaryotic and eukaryotic cells. Now, endosymbiosis is the theory that eukaryotic cells, which is the more complex cell type, most likely emerged when the last common ancestor cell engulfed smaller prokaryotic cells and coexisted with them. So basically what it's saying is the last common ancestor cell ate a small prokaryotic cell, and that went on to become what is known as a eukaryotic cell. And there's an illustration to the side showing how this process would work. So some evidence to support endosymbiosis is the fact that mitochondria, which is an organelle found in eukaryotic cells that we'll talk about today, it actually resembles a prokaryotic cell. It contains its own chromosome, it's capable of dividing independently, it has prokaryotic ribosomes, and it has bacterial membranes that are inhibited by drugs that only affect bacteria. So we have kind of strong evidence that the mitochondria is actually the prokaryotic cell that the last ancestor cell kind of swallowed up. And then those two merged together to make this eukaryotic cell that we know today. Now, chloroplasts likely arose when endosymbiotic cyanobacteria provided their host cells with a built-in feeding mechanism. So chloroplasts, we'll learn today, they're an organelle that help with photosynthesis. So photosynthetic organisms are going to have this particular organelle. So just kind of wanted to give a little more evidence of why we think endosymbiosis is a real thing. So eukaryotic organisms include things like plants and animals. However, this is microbiology, so we're only going to be focusing on eukaryotic microbes. And the main pathogenic eukaryotic microbes we will be discussing are protozoans, fungi, and helminths. Technically, algae are also considered to be eukaryotic microbes, but they're generally not too pathogenic, and we don't have a lot of time to go over everything. So we're going to focus on these three main groups of pathogenic eukaryotic microbes: the protozoans, the fungi, and the helminths. So just like with the prokaryotic cell, we had to learn about the different structures and their functions. We'll have to do the same for the eukaryotic cell. And this might be a review from previous classes you've had, but it's important that we understand which structures are characteristic of a eukaryotic cell. So in the dark green box, we have structures that are always present in a eukaryotic cell. And those include the cell membrane, the nucleus, so we're going to see a true nucleus with the eukaryotic cell, and that means it's going to have a nucleolus and a nuclear membrane, mitochondria, endoplasmic reticulum, both rough and smooth, the Golgi Apparatus, a cytoskeleton, and the cytoskeleton is made of those things that follow: the actin filaments, the microtubules, and the intermediate filaments, and then the lysosome. Okay, so those are always present in a eukaryotic cell. And if you want to see what they look like or where they're placed in the cell, you can look at this really nice figure from your book that is on the right-hand side of the screen. Now, there are some structures that are present in only some cells, so only some eukaryotes will have them. For example, a cell wall. Not all eukaryotic cells have them. In terms of microbes, fungi are going to be the primary ones that have a cell wall. Glycocalyx, remember we talked about that last time. It's going to be like this sugary coating that's outside the plasma membrane or outside the cell wall if the organism has a cell wall. So some eukaryotic cells have a glycocalyx. Centrioles, some eukaryotic cells have centrioles, and we'll talk a little bit more about what those are. Chloroplasts, which are used for photosynthesis, and then flagellum and cilia, which are used for movement. And we'll talk a little more about all these structures on the upcoming slides. So this flow chart is from your text, and it's a great way to look at eukaryotic cell structures from the outside of the cell working inward. And so we've got eukaryotic cell on the left-hand side of the flow chart, and then moving external the boundary, and then internal, and then what is associated with each of those pieces. External being at the top, the boundary meaning the cytoplasmic membrane, the cell wall, and then what's inside of the cell. So try to organize the structures like this in your mind and in your studying, and make sure that you note on this slide. This would be a great place to take notes on which structures are optional, the ones we just talked about on the previous slide that are not found in all eukaryotic cells. Okay, let's just dive right into those external structures. So this is going to be something that's outside of the cell wall or outside of the cytoplasmic membrane. So we've got appendages for moving. Just like we had last time appendages for moving, we have appendages for moving eukaryotic cells. And the types of appendages we have in eukaryotic cells are flagella, just like we saw with the prokaryotes, and cilia, which is something new. So prokaryotic and eukaryotic cells both have flagella, but they are a little bit different. The flagella that we see in eukaryotic cells are more complex and thicker than prokaryotic flagella. They're also composed of these microtubules that we see in a 9+2 arrangement, meaning that there are 9 microtubules on the circumference of the flagella, the round part, and then there are 2 microtubules in the center. And so you can see a picture of that to the right-hand side of the screen. And so this is a more complex structure than what we saw with a prokaryotic flagella. Cilia, those guys are shorter and more numerous than flagella. They're mainly for moving, but they can also be used for feeding. So where we would see, anywhere from 1 to numerous flagella, in the case of cilia, you're going to see lots and lots of cilia on an organism in the case that it's used for movement. Okay, so they're just like short little hair-like structures that are all over, and they kind of beat like wings to propel the organism around. So not all eukaryotic cells have a glycocalyx. It is an optional outer covering that you're going to see as the outermost layer with direct contact with the environment. It's also called the extracellular matrix. So this is going to be a little bit different than what we saw with prokaryotic cells. We're calling this one the extracellular matrix. And the function of the ECM is to protect the cell, to help the cell adhere to surfaces, and to help with cell-to-cell signaling. So it has a more complicated role than what we saw with prokaryotic glycocalyx. So those were the exterior structures. Let's move to the boundary of the cell. So some eukaryotic microbes are going to have a cell wall. We know lots of prokaryotic microbes have a cell wall, but not all eukaryotic microbes will. Fungi are the only eukaryotic microbes that do have one. Protozoans and helminths do not have a cell wall. So that's something important to note. The use of the cell wall in fungi is for support. They're rigid and they're used to support the cell. And fungal cell walls are quite different from what we saw with the bacterial cell wall. Instead of being made of peptidoglycan, they're made of chitin or cellulose with a thin outer layer of polysaccharides. So you can see a picture there of what it looks like, and that looks different from the figures we've seen that are describing bacterial cell walls. So not all eukaryotic microbes have one. Fungi are the only ones that do. Now, just like prokaryotic cells, all eukaryotic cells have a cytoplasmic membrane. So we can just say all cells have a cytoplasmic membrane because prokaryotes have them, and so do eukaryotes. The cytoplasmic membrane within a eukaryote is very similar to that of a prokaryote. There's going to be bilayer of phospholipids that have those embedded proteins, and it serves as a selectively permeable barrier. It's going to be letting things in and out of the cell, just like what we saw with prokaryotic cytoplasmic membrane. It's different from prokaryotic cytoplasmic membrane in the fact that it contains sterols, which are used for stability. And sterols are not something that we see in prokaryotic plasma membranes. So the addition of those sterols to the eukaryotic cytoplasmic membrane is helpful for cells that don't have a cell wall, because remember that cell wall helps give them stability, and so for those eukaryotic microbes like protozoans and helminths that don't have a cell wall, those sterols really help keep their cell stable. So we've talked about what's outside, we've talked about the cell boundary. Let's talk about the internal cell structures that we see in eukaryotic microbes. Now remember, what we're going to call a lot of these structures are known as organelles. So an organelle is a little organ. It means it's going to be a membrane-bound structure that's going to perform a specific function. So those would be things like the nucleus, the Endoplasmic Reticulum, the Golgi Apparatus, the Mitochondria, and Chloroplasts. Those are known as organelles, so they're specialized in membrane bound. So it's really important that we note that they are membrane bound, because that's not something we saw with prokaryotes. So we're going to talk about all these different structures in a little more detail on the upcoming slide, but this is just to kind of organize which structures we're going to be talking about. So you need to know all of these here. Okay, so let's start with our first two structures on that list. So remember, these are going to be internal structures. They're within the cytoplasmic membrane. So remember, in prokaryotic cells, we have this gel-like matrix that is bound by the cytoplasmic membrane, and we call that the cytoplasm. Well, eukaryotic cells have the same thing. They have a cytoplasm as well, and suspended within the cytoplasm are different organelles. And the cytoplasm is considered to lie between the cytoplasmic membrane and the nucleus, which leads us to the nucleus. It is only found in eukaryotic cells. It is a separate and distinct area of the cell from the cytoplasm, and it's separated from the cytoplasm via the nuclear membrane. And so the nuclear membrane is just going to be this envelope that surrounds the nucleus. And you can see what that looks like in the figure to the right. There's kind of a little zoom in of the nuclear envelope. So that's what separates the nucleus from the cytoplasm. There are two important parts of the nucleus that you need to know about. There's the chromatin, which is basically the DNA. It's the genetic material in the cell that's going to tell the cell everything to do. And it's suspended in the nucleoplasma, which is a gel-like matrix of the nucleus. There's also the nucleolus, which is this darkly staining body within the nucleus. You can see in the actual micrograph there, that dark staining body, that circle, that's the nucleolus. And that's actually where rRNA is made, ribosomal RNA. And that is going to be incorporated in the ribosomes, which we'll talk about in a moment. But last time we brought up that in prokaryotic cells, they are the site of protein synthesis. And it's going to be the same for eukaryotic cells. So the nucleolus is an area where that rRNA is made, and that's going to be incorporated in those ribosomes. So moving down our list of internal structures, that leads us to the endoplasmic reticulum and the Golgi apparatus. And these two organelles are involved in production, packaging, and shipping of proteins and lipids. So let's start with the endoplasmic reticulum. This is a series of membranes that are used in protein production, transport, and storage. And you can see a picture in the figure to the right. The purple is the endoplasmic reticulum, and you can see it's kind of like this network of tubes, and those are membranes. So we've got the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough is called that because it is dotted with ribosomes. So those little dots make the appearance look like it's rough on the outside. And so the rough endoplasmic reticulum is actually, it originates from the outer membrane of the nuclear envelope. So it's continuous with the nucleus, and ribosomes are attached to the membrane, and that is where protein synthesis is going to happen. Now, once those proteins are synthesized, they're transported to the ER for storage and transport. Now, the smooth ER doesn't have ribosomes. It's just kind of the passing along the assembly line. It's used to process those proteins and synthesize and store lipids, which are fats. Now, the Golgi apparatus is a separate deal. It's a separate organelle, but it takes the proteins that were made on the ribosomes in the endoplasmic reticulum, and it modifies them and ships them out. It's kind of like what we would call the post office of the cell. It's going to get them packaged and ready to be sent out of the cell. And it's a series of membranes that are similar in shape to the endoplasmic reticulum, and they work together with the endoplasmic reticulum. So the ER sends out these little transitional vesicles, which you can see at the bottom of the figure, those little purple looking bubbles. Those are those transitional vesicles. So the ER sends proteins and lipids in those vesicles, and then those vesicles meet up with the Golgi apparatus, which is represented in orange on this figure. And when they get to the old Golgi apparatus, what's going to happen is they'll be modified. They'll be modified by adding sugars or polysaccharides and or lipids. Now, once they're modified and they're ready to go, the proteins and the lipids are pinched off into condensing vesicles for use inside the cell, or they're getting ready to go to be transported outside of the cell. So the endoplasmic reticulum and the Golgi act together to produce, package, and ship proteins and lipids for use inside and outside of the cell. Okay, our next internal structure is the mitochondria. So mitochondria is the plural and mitochondrion is the singular of this organelle. So mitochondria are the powerhouses of the cell. They're responsible for generating all of the energy, or ATP, because the cell uses energy in the form of ATP. And so it's responsible for generating all of the energy for the cell, and that energy is used to carry out the different activities that the cell is going to do. Now these guys are unique in the fact that they divide independently of the cell, they have their own DNA, and they have prokaryotic ribosomes. And I mentioned all these characteristics earlier in this presentation when we were talking about endosymbiosis. And so we think that mitochondria were originally their own prokaryotic cells that were enveloped by the ancestor cell and now have become part of a eukaryotic cell. So something to point out about the structure of the mitochondria, note the interesting structure. It looks kind of like a bean, and then it has these kind of like folds within it, okay? So it's got a double membrane. It's got this outer membrane, and then it's got a folded inner membrane. And the folds of those membranes inside are called cristae, and that is where the ATP production is actually happening through cellular respiration. And we'll talk more about that when we get to metabolism. But it's important that you understand the mitochondria is the powerhouse. It's where the energy is going to happen, and only eukaryotic cells have it, not prokaryotic cells. A few more internal structures. Chloroplasts, I've mentioned a couple of times today. They are found only in photosynthetic organisms, in eukaryotes. They are organelles that convert light energy into chemical energy for the cell to use. Ribosomes are the protein factories. We've talked about those last time, and they are larger than prokaryotic ribosomes. So instead of the prokaryotic 70S, they're actually 80S. And these ribosomes just float free in the cytoplasm, or they can be attached to the rough ER, or they can be found in the mitochondria. So they're kind of everywhere in the cell, and they're making proteins. The cytoskeleton is just what it sounds like. It is the skeleton for the cell. It's a complex network of these tubules and filaments that help maintain the cell structure, that help move the cell, that help place organelles within the cell, and that help move things around within the cell. So it can move the cell itself, and it can move things within the cell. So it does a lot of things, this cytoskeleton. And it's composed of actin filaments, microtubules, and intermediate filaments. And these three different types of tubules and filaments differ in how they work to help construct the cytoskeleton. And if you want to see what that looks like, you can look at the figure to the right. That's actually a fluorescent microscopy picture of the cytoskeleton, and all of those little green fiber looking things, those are all parts of the cytoskeleton. So this is a great table that basically compares prokaryotic and eukaryotic cells, and it throws viruses in there too, which we'll get to next time. But it's good for you to kind of see side by side what prokaryotic cells have versus eukaryotic cells. So be familiar with the information of this table. We've talked about a lot of it already, but the way that it's formatted, it will help you compare and contrast these major cell types very easily. So I would be familiar with the information on this table. All right, now that we know a little more about eukaryotic cell structure and function, let's talk about those three main groups of eukaryotic microbes. So remember, those are the fungi, the helminths, and the protozoans. And we'll start with the fungi. So the good news is that fungi are major decomposers in the environment, and that's great, because if not, dead stuff would be piling up everywhere. So they are super important in terms of decomposing things in the environment. They also form really important relationships with plant roots and help them obtain water and nutrients. And these associations are known as mycorrhizae, and it's really important in terms of the plant biology and them getting the nutrients and water that they need. Another really good thing about fungi is they are a really important part of our diet directly and indirectly. So some of you might not agree with this. So if you don't like mushrooms or truffles or truffle oil, things like that, you may not be partaking directly of fungi. But most of us partake of food that has been created indirectly by a fungus. For example, bread. If you've ever eaten any kind of bread that rises, so any kind of leavened bread, then you are taking advantage of the fact that yeast can produce fermentation, and that is what helps produce bread. Bread, pizza crust, all of those yummy, delicious things. Also, beer and wine. Yeast help us with that as well. The bad news about fungi is that they cause mycoses, which is a word for fungal infections. Okay, so you might want to highlight that word. Mycoses are fungal infections, and they can be opportunistic infections in weakened individuals, and they can also be primary infections in healthy patients. It really just depends on the fungus we're talking about. So even though fungi do a lot of really great things for us, they also, there are some pathogens that we need to be concerned with. So there are two main forms of fungi. We have the yeasts and we have the filamentous fungi. So the yeasts are very simple. They're unicellular. They have a round or oval shape, as you can see in the figure below, and they reproduce asexually via budding. So basically they just bud off another yeast directly from their one single cell. Now, filamentous fungi are much more complicated. They're also known as the molds. They are multicellular, so they're going to be composed of more than one cell. This is the first time we've seen that in micro so far. And they're composed of these long filaments called hyphae. That's definitely a good vocabulary word. And so these hyphae are these long filaments that make up the body of a mold or a filamentous fungi. Now, if you'll notice in the bottom box, some fungi are very special and can take either form, yeast or filamentous form, and so we know those as the dimorphic fungi. And a lot of times this is dependent on temperature. Maybe at room temperature they exist as a filamentous form, and at body temperature they exist as a yeast. And so those are called dimorphic fungi. So an important term you need to know. Okay, so what do fungi eat? So let's talk about fungal nutrition. So fungi are heterotrophic, meaning they have to acquire food from organic substrates, meaning they're not photosynthetic. They can't make their own food, so they have to get it from somewhere else. And so where do they get it from? Primarily they are decomposers, which I mentioned earlier. And another word for decomposers is a saprobe, or we can say they are saprobic. And decomposers obtain nutrients from the remnants of dead plants and animals. They break them down by penetrating the surface of the substrate, which is whatever the food source is. We call it the substrate. And then they secrete enzymes down into the substrate to break it down and kind of suck it up. So that's what fungi do. Now, some fungi can be parasitic and live off of live plants or animals. And a lot of the pathogenic fungi, that's the case. They're parasites. Now let's look at some terms related to fungal morphology. Now remember, morphology means what it looks like, right? So in terms of filamentous fungi, we have quite a few terms we need to talk about. So mycelium is the word for an intertwining mass of hyphae that forms the body of a mold or filamentous fungus. So you've seen this before. It's just the furry, the fuzzy, all these little hyphae put together. That's the mycelium. The septa are cross walls that are found in filamentous fungi hyphae that allow the organism to have compartments. And so instead of that long hyphae just being one big cell, it's broken down into cross walls called septa. Non-septite hyphae lack cross walls. They're just one continuous cell, which we also call cenocytic. So that's an important term for you to know. Cenocytic means they lack septa, so they're going to have continuous one cell. It's going to be one big cell throughout the hyphae. We also have vegetative hyphae and reproductive hyphae. The vegetative hyphae are going to be the hyphae that run along the horizontal path. They're going to be the ones that obtain the nutrients. The reproductive hyphae are going to be the ones that extend vertically into the air, and those are going to be the ones responsible for producing spores, which is the main form of reproduction for fungi. So speaking of spores, let's talk about the reproductive strategies of fungi. So you can have asexual fragmentation of the hyphae, those filaments. If you break them off, they can make an organism identical to the original one. So that's one way a fungus could reproduce, a filamentous fungus. Another way would be through the production of spores. And we've got asexual spores and sexual spores. And these guys are dispersed through the environment. They can be blown through the air, they can float through water, they can be taken on by other organisms and deposited elsewhere, and then those spores germinate into a new fungus when they find a favorable substrate to thrive on. And remember, substrate is just going to be whatever food source they're living on. Now, asexual spores produce organisms identical to the single organism producing the spore. So they're going to look just like the fungus that originated those asexual spores. And we've got two main types, and I have included figures that describe what those types look like on the right-hand side. We've got sporangiospores, which are asexual spores enclosed within a sac known as a sporangium, and that's going to be at the top photo of the page. And then we've got conidiospores, which are asexual spores that are not enclosed in a sac. They congregate in one particular area, but they're not enclosed in a sac. We also have sexual spores, which produce organisms that are a combination of traits from two parent fungi. And this is important because it can lead to potentially advantageous adaptations. It's not looking just like one parent. It's going to kind of give some variety in the different generations. Okay, let's move on to our next big group of eukaryotic microbes, the protozoa. So these guys are single-celled organisms with all major eukaryotic organelles. So it is a single cell. They're not going to be multicellular. They're going to just be one cell. So all protozoans are single-celled. They're heterotrophic, so they're not photosynthetic. They can't make their own food. They have to make their way in the world and get the food. They have to feed. They can be free-living or they can be parasitic. The normal vegetative cell of a protozoan is known as a trophozoite, so definitely make sure you know that term. That is just the term for the motile feeding stage of a protozoan. It's just the normal cell that's going out in the world. It's feeding, it's living, it's reproducing. That's the trophozoite. Protozoans can reproduce both sexually and asexually, so oftentimes they have very complicated life cycles. They are sensitive to the availability of moisture, but they're usually able to survive extreme temperatures and pHs pretty well. But they have to have moisture in order to survive. They can convert to a resistant dormant cyst stage when conditions are unfavorable. And this is kind of similar to a bacterial endospore. A cyst, like an endospore, is very resistant to extreme environmental conditions like heat, drying, and chemicals. So a cyst is similar in the fact that it protects the organism. It can be dispersed by air currents, by water, things like that. And it's important in disease transmission because it keeps the organism safe when conditions are not favorable. So it allows organisms like Entamoeba histolytica and Giardia lamblia to be transmitted in food and water because they can survive for a lot longer outside of favorable conditions. So it's important in terms of disease transmission. But not all protozoans can form a cyst. And so those that can't form a cyst are oftentimes not able to survive outside of the host cell because those conditions aren't as favorable as the ones that are in the body. And I have an example there, Trichomonas vaginalis. It has to actually be transmitted person to person because it can't form a cyst and it will not be able to survive those unfavorable conditions outside of the body. So this table is from your text. It's just to acquaint you with some of the pathogenic protozoans we will be talking about later this semester, give you an idea of what type of diseases protozoans cause and where they are normally at, which is the source or the reservoir. And also there's a few pictures there to kind of show you what these different protozoans look like. So, okay, let's move into talking about our last group of eukaryotic microbes, the helminths. So adult helminths are usually large enough to be seen with the naked eye. Some are free-living, but pathogenic ones are parasitic. And so, of course, that's the ones we'll focus on because those are the ones associated with disease. And some spend part of their lives in the gastrointestinal tract of an animal host. A lot of them usually do. They are split into two major groups. You need to know these groups. The flatworms are also known as phylum Platyhelminthes. These guys have a thin, segmented body, and it includes cestodes or tapeworms and trematodes or flukes. You need to be familiar with all of these different terms. And you can see what those flatworms look like. They're very flat. They don't have a lot of three-dimensionality to them. They're very flat. So the flatworms are flat. The roundworms are the other group of helminths that are important. They are phylum Askelminthes. They are elongated, cylindrical, and they have an unsegmented body. They are also called the nematodes. So roundworms are also known as nematodes. They're going to have a little bit more three-dimensionality to their bodies, and they're more cylindrical, meaning they're more like a tube. So the two main groups of helminths, the flatworms and the roundworms. Okay, helminths are multicellular animals, so they're considered to be animals. They're grouped in with the animals, but they are microbial, right? They're very small. Even though adults can be seen with the naked eye, a lot of the life cycle is microscopic. So these guys have organs and organ systems, so they're the most advanced organisms that we'll look at in microbiology. The most developed organs are the reproductive tract because they're parasites, or when we're talking about the parasitic ones, perpetuating the life cycle is the most critical part of their life, is making sure that they're reproducing to get themselves out there and get themselves into other hosts. So the life cycle for all helminths includes the fertilized egg, the larval, and the adult stages. So the egg or the larva is the infective form passed from one host to another. And depending on the organism, depends on whether it's the egg or the larva that's the infective form. The host in which the larva develops is known as the intermediate host, and the host in which the adult lives is the definitive host. And you're like, well, why are they not the same one? Because like I said, these guys really want to get themselves out into different hosts, and so their life cycles are usually pretty complicated, and they're in different hosts in different places. And so it's really important you understand that the intermediate host is where the larva develops. The definitive host is where the adult helminth lives. So something that's kind of interesting about helminths is that some helminths have sexes separate and some are two in one organism. So the cestodes are generally hermaphroditic. So remember, the cestodes are the tapeworms. They are part of the flatworms. They are generally hermaphroditic, meaning they have both sexes in one organism. So you're going to have both male and female in the same organism. Trematodes, which are also part of the flatworms, they can be hermaphroditic or they can have sexes separate. It depends on the particular organism. And nematodes have sexes separate, and the females are usually larger than the males. Just some kind of important characteristics about cestodes, trematodes, and nematodes to help you distinguish between the different groups. Let's look at an example of a helminth life cycle, and we're going to be looking at the pinworm, which is Enterobius formicularis is the scientific name, and so pinworm affects humans. And so let's look at what happens. So basically what happens, you have this little girl and she swallows the microscopic eggs, because for this particular organism the eggs are infective. The eggs can come from self-infection, meaning the person was already infected, they didn't wash their hands after they used the restroom, and she has her fingers in her mouth. The eggs can also come from another infected person or from a contaminated object. So these eggs hatch in the intestine of the new host. The larvae mature into adults within 1 month, and then the adults mate and the female migrates to the anus to deposit eggs. Okay, and so the eggs are in the anus, and that's of course going to be expelled at some point, or there can just be some personal hygiene issues that allow for self-infection. Now, part of the life cycle is this intense itching that occurs in that anal region, and the scratching spread the eggs to the hands, which then touch other people or objects. And so you can see now she's passed it along to her little friend that she's playing with, and because she's been scratching in that area, not washing her hands, and then now this kid's probably going to get pinworm too. So this is a fairly simple life cycle for a helminth. Usually there's more animals involved, different hosts, etc. But just to kind of give you an idea of how these guys perpetuate themselves. And here are some common helminths, their modes of transmission, their diseases, and how their life cycles are perpetuated of some various pathogenic helminths that we'll be talking about this semester. So let's apply this eukaryotic microbe knowledge with a focus on disease. We're going to be talking about malaria, which is caused by a eukaryotic microbe. Now, malaria is one of the biggest infectious diseases worldwide. It affects an estimated 250 million people worldwide every year. So that's a ton of people. Even though we don't see any cases in the United States except in travelers, it is still something that is very important because it affects so many people all over the earth. Approximately 1 million children under the age of 5 die from malaria each year worldwide. So that's a lot of children that are dying from this disease. Malaria is caused by the eukaryotic microbe Plasmodium. It is a protozoan. I'd probably underline that just so you'll remember that. So Plasmodium is this protozoan that is spread through the bite of a mosquito, and it's geographically limited to tropical areas, particular sub-Saharan Africa. So we don't see malaria everywhere because the particular mosquito and the particular pathogen are geographically limited to these certain areas. Symptoms of malaria include joint and muscle aches, headache, malaise, fever and chills, and of course if it's left untreated it can cause death. Traditionally, treatment for malaria has been quinine, but there have been resistance issues in recent years, so artesunate is emerging as a more common treatment of malaria. Now, of course, prevention of this is going to include any preventative measures to limit contact with human skin and mosquitoes. So keeping skin covered, using repellents, using mosquito nets, staying indoors when mosquitoes are most active, like when the sun is going down, that type of thing. So that's malaria. It's a worldwide issue, and it's important for us to know it's caused by a protozoal pathogen.